Welcome everyone to today's Beyond the Scope discussion series. Before we get started, I want to just mention the upcoming events that we have going on. So the next event and last one of the semester, we'll be looking at some different features of the Heloscan micro CT with Carly Goodwin. Then we'll have our holiday break over December, and then moving on to next semester. The schedule is still somewhat to be sorted out, but there are possibly additionally seminars and workshops with Gatan and EDAX with a couple days on EBSD and some of their new technology and some time there, along with working on some cryo and EELS workshops as well. So those will be things we'll be working into the schedule as well. Today, we have Yoshie Narui giving a talk in the cryo electron microscopy field, kind of understanding how to do some single particle analysis with the CryoSPARC software. So I'm excited to hear about that a little bit, some of the technology that's going on there. And again, all these talks, we try and keep them around 30 minutes or so. So there's plenty of time for questions and answers afterwards. So feel free to post questions during it. We'll get to those probably at the end, but post as many as you want during it. We'll talk about those. Or if you have any other questions, concerns during the webinar, we'll be monitoring the chat as well. Okay, Yoshie.
Okay, thanks for the introduction. So as Dan mentioned, I'm part of the Cryo-EM team here at CEMAS. And so as a preface, I'm just gonna go ahead and say that the target audience for this talk would be people that have not ever done any data analysis, single particle data analysis before. So if you've used CryoSPARC or rely on before, I'm not sure how terribly helpful it'll be, but this is meant to be purely an introduction for beginners. And I'm gonna go through a tutorial that CryoSPARC has put out, but I think it's useful to kind of walk through that with people so people have an idea of what's involved. I'm going to be switching, I'm just going to stop my video, but I'm going to be switching between my presentation and then actually showing you in CryoSPARC how to do certain things. Okay, so just as a reminder, there's lots of CEMAS resources available. So obviously you're attending this webinar right now, but we have lots of other instrument managers and experienced microscopists here who can help you. So I just highlighted here my email if you would like to reach out and learn more about Cryo-EM here. We always do this at the beginning of every webinar, but we just wanted to let you know about the instrumentation that's available at CEMAS. There's a wide variety of TEMs. Specifically, in this case, we would be talking about the Glacios and the Cryos, where you would collect high resolution data. So that's why that's highlighted in red. But we also have some dual beam, focused ion beam, two of those instruments, as well as SEMs that are available as well, along with some other analytical techniques. So if any of those are of interest to you, please reach out and we can get you connected to the right person. We always also like to do a little bit of background information about who's giving this presentation. So just as, I'm not sure if you came to my first talk, but I grew up in Ohio and I was actually an undergrad here at Ohio State, and I have my BS in chemistry. I moved to Pasadena and got my master's degree at Caltech, focusing mainly on nanomaterials and atomic force microscopy. And then I moved to Atlanta, Georgia, where I got my PhD in biomolecular chemistry at Emory University. I was studying the Notch activation pathway. And then finally, I came back to OSU and started my postdoc in the Sotomayor lab, where I learned X-ray crystallography and ran molecular dynamic simulations, all as part of a project related to human hearing and mechanosensation. And so finally, I ended up here as sort of a structural biology expert at CEMAS. Okay, so to cover just very quickly, I'm only going to be talking about sort of single particle analysis and how to treat the data, but we offer a wide variety of cryo capabilities, including tomography, micro-ED, and we're also building a cryo-fib milling capability as well. So those are all things or projects that we could help you with if you're interested in. So not just single particle analysis, but that's the main focus for today. And just as a brief reminder, I'm sure everyone here is aware of how influential Cryo-EM has become in the last 10 years, especially for structural biologists, and the advantages of this technique versus, let's say, X-ray or NMR. In many ways, they're complementary, but for Cryo-EM, you can look at your sample in a native state. You don't have to grow crystals. It doesn't require a huge amount of sample. But like any technique, it does have some limitations in the fact that the size of the molecule has to be relatively large. So at least probably around 100 kilodalton or larger. And then also you have to be very aware of how to treat your sample and not to expose it to too much radiation and things like that. So that's just something to keep in mind in terms of whether or not your project would benefit from Cryo-EM. And as a reminder, the workflow, this is a very simplified sort of chart, but it always begins with good biochemistry where you have a nice clean sample, and then you screen by negative stain initially, and then move forward with your Cryo-EM sample prep. Sample optimization can also take quite a bit of time, but we do have the Glacios, which can help you screen samples. And then finally, the Krios can be used for data acquisition. So the question becomes, right, after, let's say you've gone through all those steps and you have your data in hand, now what comes next, right? So in terms of software for Cryo-EM image processing, you're gonna have a lot of choices. And I'm only gonna talk about CryoSPARC, so that's just one of the many options that you have. All of these, I believe all of these programs are freely available to academic users. So you should probably test a couple of them out, see which ones work best for you and sort of how you process data. I will say for most of my users or many of my users, they do prefer CryoSPARC, especially when they're starting out. It just seems to be a little bit more user-friendly for them. And so I would say Relion and CryoSPARC are probably the two most popular choices. But again, there are many to choose from and each has their own advantages and disadvantages. Okay, so to follow up with that, I'm gonna be talking, like I said, specifically about CryoSPARC. CryoSPARC is an acronym. Here you can see it's for Cryo-EM Single Particle Ab Initio Reconstruction and Classification. And it is a web-based software. So the GUI is essentially something you would open in Firefox or Chrome. And this workflow here, this chart kind of shows you all of the steps that you'll go through after you've collected your data. So as you can see, it's not perfectly straightforward, but I'm gonna walk you through sort of what's called the pre-processing steps, which is motion correction, CTF estimation, and curation. Then we'll move to particle picking and 2D classification, and then finally to the ab initio 3D classification or model building to get your final structure. I'm not going to be spending much time on this bottom right corner, which is sort of more advanced refinement of the maps. So sharpening and local refinement, or if you have like a very flexible or heterogeneous sample, there's really not time to cover that, but that's definitely something that people do to improve their maps further. If you want to take the time later on to go through the tutorials yourself and take more time to carefully go through them, I mainly use these two links that you'll see here. So the one is hosted by CryoSPARC for the proteasome data set. And then the second one is a tutorial that's been put together by NCCAT, which is one of the national cryo-EM centers, but it has some very detailed instructions that will also be helpful for you. Okay, so let's just talk a little bit about CryoSPARC and how it works. So CryoSPARC has sort of a hierarchical structure where you have projects that are sort of standalone. So you can't talk between projects necessarily. So project would be a specific sample. It doesn't, it could be a complex, whatever you want. And then within that project are individual workspaces. And so these workspaces could become, or could come from separate data collection times. Maybe you prepared your grid slightly differently. So you can put all of these in different workspaces. And then within the workspaces are individual jobs. And these jobs are basic processing operations. For example, like motion correction or CTF estimation. And the jobs are numbered so that you don't get an overlap, because you can actually link together jobs between workspaces. So here you can kind of see how they're numbered, and I'll show you how you actually can link them together. Okay, so that is kind of to give you an idea of how it's structured within the program. Okay, so I'm gonna go ahead and try and switch here to CryoSPARC and show you the dashboard and a couple of important things to point out. So hopefully technology works here. Okay, so just wanna make sure, hopefully everybody can see the dashboard now. So this I've just opened in Firefox. Okay, so there is a computer here at CEMAS that's running the software that's running CryoSPARC, a Linux machine, but I use a web browser to access it. So that's this address here. Okay, so essentially what the dashboard is going to tell you is a summary of all the projects and workspaces and jobs that are currently being run or in total that you have. It also gives you links to the discussion forum if you have questions, and also the databases that hold Cryo-EM data are available to you here. So you'll notice here at the top, there's what I just mentioned, projects and workspaces that are available. If I click on the drop-down menu, you'll see all the projects that have been created. And so what I'm gonna do is I'm gonna click actually on this drawer here, and it's gonna sort of graphically lay out all of the projects that I have. I'm not gonna create a new project, but you could, if you wanted to, just add a new project. I've already created one for the proteasome tutorial data set, which is this project 18 here. If you wanna enter the project, you just have to click on the square that said P18. Okay, so now I'm in the proteasome project, and it has at least 2 different workspaces here. So you'll see there's workspace 1 and 2. So in workspace 1, I've already gone through all of the steps that were outlined in the tutorial. And then workspace 2, I've just created as a live demo. You'll notice that all of these jobs, so you see all the jobs are numbered and color coded. And green essentially means the job has been completed. Red means the job has failed. And then purple, or this bluish purple color, is either you're in the process of building the job, and then or it's running currently. So these are just not complete yet. Okay, so here are all the jobs and workspaces that you can see. And if you look over on the right-hand side, you'll notice that there's a job builder off to the side. Everything's grayed out because I'm not currently in a workspace. So it's telling me I have to pick a workspace to actually create a job. So just for illustrative purposes, I'm gonna add a new workspace, and I'm just gonna say it's test here and create it. Okay, so I just made a 3rd workspace here. So I'm gonna enter that workspace by clicking on that box. Okay, so now I'm in this test workspace, and you'll notice that everything in the job builder became available to me. So they're not grayed out anymore. Okay, so you'll notice that it's broken down essentially into different job types. So the pre-processing would be importing the movies, motion correction, CTF estimation, that's what's considered pre-processing. And then also you'll notice some of them have this picture of a monitor next to it. Those are all interactive jobs. So at some point we would have to do something in that job to get it to run. And we'll do an example of that with the manual picker in just a little bit. But everything is available on the side here. So it just goes in the order in which you would actually carry out the jobs. So from 2D classification to 3D refinement, and then it gives you some extra post-processing options down at the bottom. Okay, so let me just switch back real quick to the presentation. Okay, so I just went over the hierarchy. As I mentioned, the first 3 steps of any data analysis is going to be actually importing your raw movie files. Then you're going to do the motion correction, which is going to align all those movie frames together and average them. And it's going to essentially de-blur the movies. If there is any stage or sample movement, it will correct that. And then the last step is going to be patch CTF estimation. And so that is actually going to try to measure additional parameters like defocus and stigmatism that vary from micrograph to micrograph. Now, I'm not going to go into the math or the details of all of this because that would also be another webinar. I just want to say something real quick though about CTF estimation. So for those non-microscopists, the contrast transfer function is probably a little bit of a foreign concept. So I'm kind of trying to make a more practical definition. So I'm just going to give you like 10 seconds to read these couple sentences. Okay, so practically speaking, the contrast transfer function, if we look at the 2 images that are on the right here, these are both carbon films, thin carbon films that are taken at different defocus values. So the one on the left was taken at minus 0.5 microns and the one on the right was at minus 1 micron. So if you look at the images underneath, these are the FFTs of the images above them, right? And you'll notice the tone rings here. The plot in white is the actual CTF plotted out, overlaid on top of the data. And so this is essentially what it's estimating from the images that you provide. So the one thing that you should notice, right? So especially this sentence here where it says, you're trying to recover obviously the high resolution information. And you'll notice that the frequency of the oscillation increases with resolution. So what does that mean? So that means the closer you are to focus, actually that's where the highest resolution information is going to come from. But the issue is the closer you are to focus, the less contrast you'll have in your image. So it'll be more difficult to select particles. So it's a little bit of a trade-off. But what CTF estimation is actually doing is taking your image and then essentially calculating a function that will match your CTF. Okay, so I'm not going to show you the equation, but you have questions about that, we can cover it at the end. Okay, so what else do we need to know? To import the movies for any data set, but specifically for this tutorial, you need this information. So this data set was deposited into Empire, which is one of the databases for Cryo-EM data. It's just the first 20 movies, so it's not the whole data set. But you do need to know certain conditions of how the data was collected. So you need to know the accelerating voltage, spherical aberration, pixel size, and dose. And that information should be provided to you either by the center that collected the data, or you should have recorded this yourself. Okay, so now I'm going to actually sort of walk you through the pre-processing steps. Okay, so we have all these details about the data, and now we have to go back to CryoSPARC and actually import that. Okay, so as I mentioned, the jobs are all located over here to the side. So the first step would just be to import the movies, right? So you would just select import the movies. And here it gives you a bunch of information that you need to provide. The first is obviously the location of the movies. So I'm actually already in the right folder. I'm just gonna tell it to look for the TIFF files. Okay, so I just selected all the TIFFs that were in that folder, and then we need to provide a gain reference. Unless the data was collected gain normalized. If that's the case, then you don't need to supply the gain file. But in this case, it's not gain normalized, so I need to tell it where it is. So that's the gain reference file, this MRC file, and I need to select. So if you kind of hover over these, it does sort of provide some more information sometimes. If it's green, it means you changed it. If it's blue, it's the default number. Sometimes you do have to flip the gain reference, and that's indicated here. You can do that, but that's not necessary for this one. Additionally, so as I mentioned, all those details that I had listed out on that slide, now is where it becomes important. So I actually have to type in here the raw pixel size. So I'm just going to do this real quick. And then the accelerating voltage, which is 300 kV. I have to provide the spherical aberration and the total dose. Okay, you can also provide negative stain data as well, but you'd have to select that here. And so I'm just going to go ahead and queue, which means I'm going to submit this job. I'm not going to go into explaining the lanes, but I'm just going to submit the job and let it run. So what you'll see here is that it's going to actually read in the information from each of the files. And then you should see the image actually appear here. And you'll notice it gives you this message that that job is actually running. Even if I close this window, right, I can still see that it's running. Closing this job window does not cancel your job. And if you want to reopen the job, you just have to click on the box and then hit spacebar and it'll come back. So here you can actually see the raw images as they come in. And then here a message pops up when it's done. Okay, so that's fairly straightforward because it's just reading in the images. Okay, so the next step in pre-processing, as I mentioned, would be motion correction and CTF estimation. Okay, for that, those are a little more time consuming. So I'm actually gonna move to the workspace that I had created here. So the same, job one is the same, I just imported the movies. The next step would be patch motion here. So what I can do is I can click on this job and actually link the job to the other workspace. So now I can go here and I have this information available to me. So it's right here. It's inverted because it's just going in the total, like as I do the jobs, if that makes sense. Since I already ran this job earlier, it's labeled job 3. So it just flip-flopped them. But here's the patch motion. Okay, so when I ran this job, I'm just gonna open it. You can click on here, you see the tabs. What did I input when I tried to build this job? Essentially, I just did the default for the patch motion. And what you'll notice is it shows you a plot for each image. And instead of doing motion correction over the entire image, it just did it in smaller patches. So you can kind of see how how portions of the frame move over time, right? So actually in the center, there's very little motion, but towards the edges, there was a lot more motion. And then you can literally see that for every single micrograph. I think it only does it for the first 10, but you can specify which ones. And in some cases, you'll notice that it will improve over time. So this one is probably not as bad. And then that gives you the motion corrected image underneath. So if you kind of notice, if you were observant and looked before, the imported movies were a little bit blurry, but these should be sharpened now. Okay, so that is patch motion. And so the way I would have built that job is I would have come over here and I would have just selected patch motion here. And then I would have, now the easiest way to import the movies, so I'm just gonna pretend like I didn't show you the final result there, but I would have just opened the movies that I imported. So here it says imported movies. I can literally just drag and drop the data in there. And I don't change any of these other parameters because I did everything as default. And then I would queue the job. And that's all that I would need to get. And I would end up with the same result that I have in this job here. Okay, so similarly, so I'm just gonna cancel this. I could build another job. So the next step would be CTF estimation, right? So that's this patch CTF estimation here. And you'll notice it's asking me to input the information here. So it wants the result from the previous step. So it wants the result from the patch motion. So now I can just open the patch motion job here and I can just drag and drop the micrographs in here. And I probably would not change anything from the default settings again. And then I would just queue it and run the job. Okay, so those first two our first 3 steps are pretty much using the default settings that CryoSPARC provides. And that's usually sufficient to get going. Okay, so I'll try and get to the questions at the end. So hopefully they'll all come at the end, but I'm just gonna go back to the first original workspace. Okay, so if I wanna look at the CTF results, it's not particularly exciting because it's actually very clean data. So it's just like with motion correction, it's calculating a CTF estimation all across the image. So the defocus might be different across the image, but actually this image shows that it's very consistent. So it will provide this information for the first 10 micrographs, I believe. Okay, so that's all pre-processing. Okay, I'm just gonna go back to the presentation now. Okay, so those are pretty standard, I would say, that part of the workflow. There's not a whole lot you would vary. The next step is you have some more choices of how you want to do things. You're gonna start picking the particles from the images at this point. So you wanna successfully pick all the particles, but no junk particles. So nothing that might kind of look like a particle, but really isn't. And there's a couple ways to do this. The way the tutorial does it is that you actually manually pick some particles first. So you pick about 100 particles and you create an initial average. And then you use the template picker to pick them automatically for you. That is one strategy. Another strategy is if you don't really know anything about your particles, you can do what's called a blob picker. So you give it some general information about the size and shape, and you tell the software to just pick everything that looks like that. So it's good if you don't know anything about your sample, but it can also tend to pick lots of different things that aren't your sample. And I would say the one kind of getting the most momentum are these deep learning algorithms, which are available in Warp or Cryolo. Topaz is actually integrated into CryoSPARC, but in the deep learning algorithms, you have to train the particle picker, and then it learns which ones are just noise and which one are your particles. And then it actually does a better job than you would do by eye. So these are actually getting quite good. And I would say a lot of people prefer to use something like Topaz. But in the tutorial, we will do the manual picking and then the template picker followed by that. And so then once you have picked all the particles, then you essentially need to extract all the particles out and then you classify them. So you put them into distinct groups and then you average all the particles within each of the different groups that it has made. Okay, and then that information will feed into the 3D model. Okay, so let's go back. Okay, so this is the manual picking process. Actually, what I'm going to do now to kind of make things a little bit simpler is I'm just going to click on this job 5, which is the manual picking job. And what I can do is I can just clone this job. So if I come over here, you see a bunch of actions that you can do with the job. I'm just going to click on clone job. Okay. And down here, job 28 is identical to job 5. Okay. And so it's taken all the same information and it's going, and it just creates an exact copy. Okay. So I'm just going to hit the Q button here. And again, I'm just going to pick, I'm not going to get into this, but I'm going to pick one of the lanes. Okay, so manual picker, if you looked at the job, it had a little monitor next to it, which means it's interactive. So you'll notice there's a tab here that has appeared that says interactive. So I'm going to click on it. Okay, so over here on the left, you'll see a list of all the micrographs that are available. So there should be 20 in this particular data set. And if I click on any of them, it will update with the correct image. Now you can categorize these any way you would like to. So if I click on defocus, it will put them in from the highest defocus to the lowest defocus. I usually like to start with high defocus when I do particle picking because I have the most contrast and I can pick particles relatively easily. We'll just stick with the default box size. Actually, I think in the tutorial, what they recommend is a box size of 384. Generally speaking, the box should be roughly 2 times the diameter. That's just kind of their recommendation. So now what I want to do is if I click on this, if I click where I think I see a particle, there'll be a circle that appears. Okay. If I think I picked a wrong particle, I can just right click and erase it. So the strategy would be to go through these images and pick roughly 100 to 200 some particles. And you want to try and pick ones that are different views. So clearly some of these are top-down views, some of them are side views. So you want to just literally pick through these images and you want to spread it out. So it tells you how many picks that you've made already, but you want to do like, let's say, 20 or 25 from several different images. Okay. So you would just literally go through and try and center the particle in each of these. And then when you're done, what it's going to do when you're all finished, you would just say, I'm done picking, extract the particles. So it's just extracting the location of those particles. Okay. So I hit done and then it basically spits out the results. Okay. So it says it's completed. Okay, so I have done that here already in this manual picking job. And I think what came out here, you can see on the right, I picked 198 particles. Okay, so that's the particles I picked from that previous job. Okay, so now I'm gonna take those and use those essentially as a model for the template picker. And the way I'm going to do that is I have to run a 2D classification job. So I picked 200 particles randomly, right? And now I'm actually going to classify them. So I'm going to clone this job here. Okay, so what did I put in this job? You put all the particles here. So that's all 200 particles. And I'm going to tell it, I want it to classify it into 10 classes. Okay, so it's going to try and bin each particle into 10 different classes. And that's it. That's all I changed. Normally the default, I believe, is 50 2D classes, but that thinks it's like an entire data set. This is a relatively small data set, right? Because I manually picked each particle. So I'm not going to pick like 10,000 particles. I'm only going to pick a couple hundred. Okay, and then I would run this job. And the results from this job are what you see here in this 2D class here. Okay, so these are the 10 classes I get. Just from the 200 particles or 198 particles that I picked. You can clearly see here, I would say there's like 4 good classes, and then these are all just garbage that you don't want to pick, right? So actually in the tutorial, what they recommend is just to pick 2 of these 4 classes. So I believe I picked this first one here, and then this second one. So it's clearly a side view and a top-down view. I will say for more complicated samples, it could be very difficult to decide which classes are real and which ones are not. So you may indeed have to pick more particles manually. So we're gonna just reduce it down to 2 classes, okay? And the way that you would do that is running a job called select 2D. So of these 10, I only picked these 2. Okay, so if I look at this job, all I literally did was just pick these 2 and it made it these 2 classes. That's it. That's the whole purpose of this job. So now I have these 2 as my template essentially. Okay. So now we can finally move to a template picker. So obviously you don't want to pick all the particles manually because that would be a giant pain. So we want to make the process automated, and that's by running a job called template picker. Okay, so I cloned the job here, but what you can see is you give it the template, so that the template is the 2 particles here, right? So these 2 particles, and then you give it all of the data that you have. So all 20 exposures go into this box here, okay? And you do have to provide some information about the size or the diameter of the particle. So in that case, I said, okay, the particle is probably around 200 angstrom, so 190, because I know some information about my particle of interest. So let's just say we'll give it some wiggle room, so it's probably actually closer to 150, but I'm going to say 190 here. And then I run this job, and what it shows you is, I'll show you the output, is that based upon the templates that I gave it, so those 2 classes, now it has gone through each of the images and actually picked the particles itself. So I did not pick any of these particles where the dots are. That's what the software picked. Okay, so it's nice because it's very unbiased in the sense that you're not picking the particles. The software is picking them for you. Okay, so now it's picked all the particles, right? And you do have to go through kind of a cleanup step. So if you'll look a little more closely, you'll notice that there are some, like for instance, over on the left-hand side here, well, you'll notice that there's like a red dot, but there's no particle. So it's not perfect, and you do have to clean it up a little bit. Okay, and so I'm going to skip over that step just for the sake of time. But essentially, what you go through is you will threshold it and pick particles that are real. Okay, and then I'm going to clone this job here. So once it has identified particles from the template picker, then you actually want to extract all of those particles from the images. So how do you do that? Then in that case, you have to run a job that's called, and I'll show you here. So under job builder. So all I've done here is we've done the manual picker. You just kind of work along this workflow, and then we did a template picker because we don't want to pick each particle individually. This is a visual inspection. I'm skipping over that step, but you would essentially just clean it up and make sure it's actually picking real particles. So the last step is actually extracting the particles from the micrographs, okay? And that's what is this job right here, job 31. And then the input, so the parameters that, so it says I'm still building it here, which I am. Oops, sorry. Okay, so this job, essentially all we need to give it are the micrographs and then the particle locations. So the template picker found all the particles, and then we have the micrographs. So now it's just gonna go through and pick them all out. And what it will look like, it takes a little bit of time. Actually, I don't think it actually even gives you a visual of it. But if you look over here, what you can see is we started with 20 micrographs here, taking 20 micrographs, and then it picked all the particles. So you can see the total number of particles it extracted was 10,960. So from those 20 images, now we're left with about 11,000 particles. Okay. So now what does it do? So the last step of this part of the process is actually doing the 2D classification, right? So that's this job 11 here. Okay, so we already ran a 2D classification, if you remember, from manual picking. And it's basically the same, except instead of using 100 particles, you're gonna use all 11,000 particles. So you input in all 11,000 particles, and then you tell it, I told it to make 50 different classes. Okay, so if we take a look at these classes, immediately you can tell which ones are no good, right? And if you look underneath, it gives you some information about the resolution. You'll notice that these are actually ordered, I believe, from highest resolution to lowest resolution. But you'll notice it kind of jumps in between these two from around 8 to 18. So what I would do is actually just select these first 17 or so classes, okay? Clearly they're very similar. Some are side views and some are top down, but we just wanna select everything that we think is a real particle, okay? So this is kind of giving you the overall view, but the main thing is you wanna get rid of all of these particles that are kind of, are obviously garbage or junk particles. And so that's why you need to do 2D classification is to clean up the data. Okay, so if we go back to the presentation, so the final result, I'm gonna skip over the box size details, but we can talk about that later. The results from the 2D classification you see here, and you just wanna, like I said, be careful. This is a very clean, rigid sample, but for trickier samples, it may not be as clear which are real classes, okay? So that's where you have to be very careful doing 2D classification. Okay, all right. So this is like a speed version of data analysis. But okay, let's say we've narrowed it down to all the clean 2D classes that we have. There's no junk particles, there's no background noise, anything like that. So then the final steps are going to be to actually build a 3D model, right? So the first step is actually to build a low-res map, and then you take the low-res map and then you refine it to higher resolution, which would be under homogeneous refinement. Okay, so in homogeneous refinement, if you have some information about the symmetry of your molecule, you can input it at that point in time, but you have to be very careful that you're correct about the symmetry. I would say most people start with the default, which is C1. But if you know it's octahedral, if you know for certain it has a certain symmetry, you can apply it during homogeneous refinement. Okay, so let's go back here. Okay, so these are the 2D classes. Same way I would go through and actually select just the 2D classes that I am interested in. So that was what this next job is. So I went through and I just, out of all of those 50, I picked these 17 classes, okay? And now I'm gonna move forward with these 17 classes, and I'm gonna build an ab initio model here, which is job 13. So you would come over to the job builder and you would come down to 3D reconstruction and you would click on ab initio, okay? And the input for ab initio would be the previous job, so job 12 here, which is the 2D classes. So of the 11,000 that we started with, we're left with about 10,000, right? So these particles would go in here. And then you essentially tell it how many models to build. So I'm just going to do one, have it build one model, and you can specify sort of the resolution that you would anticipate that you could reach. This is supposed to be a low res map, right? So you kind of want to keep these numbers a little bit higher. 12 is a little bit high, but you could reduce it to maybe like 8 or 10 or something like that. And so then you can run this job. So this job has already been run here. And then this is the low res map. So these are just different side views, right? And then this is a top down view. It's not supposed to be super crisp yet. And this plot here, so it always gives you a lot of information. This plot is telling you about the orientation. So how many different views are you getting of your particular molecule? And if you have a preferred orientation problem, what you'll see is that you'll only get a hotspot in one, maybe likely one area. And it's just a histogram telling you how many images are at these different orientations. A lot of times people like to include this, especially if they have a preferred orientation problem, to show that it has been fixed, or that they have sort of like a complete view of their molecule. Okay, so this is the low res map. It doesn't look particularly impressive yet, but we don't anticipate that it would be. We have to do some refinement of this map. Okay, so the next thing we would want to do is actually run a different job. So if we go back to the job builder, I'm going to cancel this one. We just did the 3D reconstruction. And so now you want to do 3D refinement. So you're going to do the homogeneous refinement job here. Again, you would input the same particles that you did for the original model. So that would be whatever was in job 12. You'd stick it in here. And then the volume, so this is actually where you put in your ab initio model. So your low res model you have here, right? And you want to actually deposit it here. So that gives it a starting point to do the refinement. And then, as I mentioned before, if you know something about the symmetry, you can insert it here. I believe the proteasome's D7 symmetry, so you would put D7 here. And then you can adjust all the other parameters as you see fit, and then you would run this job. And so the final map that you end up getting is something that looks like this. Okay, so you'll see it again, real space slices from different views, so side view, top down. And then this is just the masking, and it kind of gives you the final resolution information here. Okay, so it's actually really hard to see the map this way. So what you need to do is you actually have to download the map. So the map here, you would want to download this map sharp. So that's actually going to give you the MRC file for this particular map. And it usually downloads it wherever your browser will download files. So I've already downloaded this map, and you need a different program to really visualize it. I typically use Chimera. I think other people can use different programs, but this is the final sharpened map here. Obviously you can do further refinement, but this is a 3 angstrom, roughly 3 angstrom map. And I'm trying to get my mouse back, sorry. Okay. And yeah, you can see clearly secondary structure. There's a chance that the map has the wrong handedness, which means that you would need to just flip the map. But you can very clearly see side chains, secondary structure, and everything at this point, because it's pretty impressive that on 20 micrographs you can get a map that looks this sharp. But again, it is sort of tutorial data, so it should be good. Okay, so like I said, for visualization, you would use a different program. Typically, Chimera is the program of choice. You can adjust some of the parameters in Chimera to make the map look a little bit more clean. And at this point, you could also, if you had an atomic model, you could dock it, or you could start to build a model for a new structure. If you don't know any information other than the sequence information, you could, again, start with model building into the actual map that you have. Okay, so I skipped ahead and we did that. Okay, so that's the final map. And then this is the resolution information. So pretty close. I mean, the paper said 2.8 angstrom, so I'm sure it was some sharpening and other refinement steps that I'm not going to talk about. We could have achieved that, but pretty impressive that we can get it with just 20 micrographs. Okay, that was like a very, I know, fast go through of that. But hopefully you get a feeling for the workflow. I would say sometimes what happens also, I've heard this trend happening, is that people actually, if I go back real quick, sorry. If we go back to the original workflow here, sometimes what people have been doing is actually, once the particles have been picked, you can move straight to making a model. You don't necessarily have to do 2D classification. And as I mentioned, it can be kind of dangerous because you may be throwing away data that's useful. So some people have just gone with these particle picking algorithms, and then they move straight into building the 3D model. That's definitely an option. But these are very, they're not rigid, so you can do these in sort of various orders. So you kind of have to figure out what works best for your data. Obviously, we're working with perfect data, so it looks great, but you're going to run into a lot of issues if you have a flexible protein or complex, things like that. But all of these processes in the end can help improve your final map. Okay, so that was a lot of information. I think I'll open it up to any specific questions of things that I may have just quickly gone over. I don't want to go over time, so I want to leave time to answer any questions that people might have. about CryoSPARC or data analysis.
Thanks, Yoshie, for the talk. I enjoyed hearing a bit more about the data processing that we can do at CEMAS and everything that you've been working on. It's kind of cool to hear about. So we have time for, if anyone has questions for Yoshie right now, you can go ahead and type those in the chat and we can start digging into this a little bit deeper. If you have any in-depth questions, that goes into, we can also enable microphones as well, so we can chat that way, or you can also email Yoshie and CEMAS directly, and I know she's happy to interact with you guys as well if you have a lot of questions that way.
That was a very quick fly through, but I will say CryoSPARC is pretty intuitive, so you should try, everyone should try Relion too, because I think Relion's very powerful and other with certain aspects of the data processing. You can also go between CryoSPARC and Relion. There are Python scripts that allow you to take the output from CryoSPARC and then input that into Relion if you want to. But those, it's not totally seamless, but there are ways to do that. Okay, somebody asked. Oh, yeah, it is good. So the data is obviously freely available to download. I believe you could just install CryoSPARC on your local machine. You don't, as long as you have a GPU available, it's fine. And we will make we'll make this presentation available online afterwards. No one had questions.
Hey, Yoshie, I was wondering, so all this data analysis, it's in on just a browser, right? So
yes.
There aren't really any requirements for actual physical PCs or anything for it. Where's all the data processing actually occurring and whatnot, or is there actually other requirements for it as well?
Yeah, so there's so the machine, our GPU processing happens like in the back room. It's like hidden in there, but we have an HGPU machine that is essentially divided into four GPU like lanes. So I didn't go over it, because when you submit the jobs, you essentially select a lane in which to do the processing. So you either pick 4 GPUs or 1 GPU, or if it's CPU intensive, you can specify CPUs. So it kind of depends on the job, which lane you would pick, if that makes sense. So we have built our own system that it's a Linux-based system that runs CryoSPARC and Relion and has a lot of storage space and 8 GPUs, right? So these types of systems, I think most PIs that are in Cryo-EM have in their own labs, right? Because that's, you need it to process the data. But for a smaller data set like this, you could do it like on your local machine. Does that make sense? But if you're doing like hardcore, like thousands of micrographs, you're gonna need like a pretty amped up machine to do that. Somebody asked, does our center accept proteins and samples from other universities? Yes, absolutely. We have a fair number of external users. You just have to have a dry shipper to get your sample here. There's a bit of paperwork that goes behind that, so to accept your samples. And then we discuss sort of the scope of the work, how much screening you want done, how much data collection you need. And that's all done before we accept your sample. But yes, we do accept external samples. I think I answered that. One new message. Oh, that's from Ashley. Yeah, so the recordings are posted on our website afterwards. No one wants to ask a question? I was just gonna talk, okay, so I have a couple minutes. I'm gonna go back and for orientation maps, would the map with several hotspots be acceptable when the resolution kind of meets our needs? Yeah, so for orientation, I would say, oh, sorry. Okay, now there's two questions. For orientation maps, right, so you want to have enough coverage. I think the person is asking about, let's just go here. Not that, sorry. Oh, I think it's, oh yes, here, okay. So, right, you want to look at the final model that you've built, and what you really want to see is, I mean, this one's not terribly great, but you want to see coverage over sort of all elevations and azimuthal angles. So this does cover a broad range, but if you get some that are like, if you have a preferred orientation problem, you're going to have very local hotspots, like tight spots, and you're not actually going to be able to even build a model. So I would say it depends on your data, right? So if you had several hotspots, would it be acceptable? It depends what your model looks like. That's what I would say. So you'd have to look at the data more closely. There's no hard and fast rule. I've also seen cases where people have a preferred orientation problem and then they try and fix it, and then they get a different orientation problem, but that actually is enough information to build a model, right? So like, it may not be a problem if you can get it in different or stuck in different orientations. On CryoSPARC, can it be determined if the gain reference correction has happened correctly? Yes. So it's very clear when the gain reference has been either not like flipped correctly. You'll get these weird artifacts in the images. So you'll maybe see like streaking, or the illumination looks very uneven. So it'll be pretty clear when your gain reference either was not applied correctly, or was flipped in some fashion. So you should look at the data closely and make sure that it looks good. So that's why, for example, let me just see. Right. So if you look as the movies are imported, you want to see a nice flat background. I mean, these are a little bit blurry because it hasn't been motion corrected, but you can clearly tell the gain is applied correctly. You could do a little test and like flip it and see what happens. Probably my guess is that it would look very strange, but you should know right away. Okay. One other thing I was going to mention, which I had a slide about, but I skipped over. So there is also this issue that I kind of skipped over quickly. But when you extract the particles out of the images, out of the micrographs, you have to be careful about what size box, so how much area around the particle you're actually selecting, right? Like a quick rule of thumb is that you want it to be double the diameter. But the issue is that the bigger that this area becomes, right, the more computationally expensive it is to extract the particles. And so it just will slow down your analysis. But you want enough information that you're getting the most high resolution signal from your particle. So you have to there's like a careful balance. So this equation was in the NCCAT tutorial data. So this is like a back of the envelope type of calculation, but you can estimate what that box size should be. So you don't want it to be too tight, but you also don't want it to be too big because then you'll spend your computational resources on a bunch of area that doesn't matter. Okay, that was my point about that. Any more? Getting in different directions And I would say Relion also has a very good tutorial as well. So I'm only showing the CryoSPARC one, but I mean, I guess, yeah, you can try both. They use a different, I think they use Beta Gal in their most recent tutorial, but try them both. Okay, I guess I should leave it on this last page. If you want to reach out, upcoming events, and this last slide is just to give you links of how to stay in touch with us. And reach out on Twitter because everyone in the Cryo-EM world is on Twitter, so join us.
Thanks, Yoshie, for the talk. Yeah, it's always great hearing more about what we can do at CEMAS and what some other people do that are completely different from what I do. So definitely enjoy that. So thanks again, and we'll see everyone in 2 weeks again. Thanks.